Hello, bachcho. Let's see the question. So the question says, which of the following is the least likely to be involved in stabilizing the three-dimensional folding of most proteins? So we have to identify the type of bond which has the least chances of being present in the three-dimensional or the tertiary structure of the protein. So the key concept over here is to understand the 3D structure of proteins. Now, the 3D structure of proteins are basically known as the tertiary structure of proteins, and they are formed by the interaction of the R groups present in the amino acids of the proteins from which it is formed. So depending upon the type of the R group participating in the bond formation, that will decide the type of bond which is being formed. So tertiary structures are formed from the secondary structure of proteins by the various types of interactions that takes place between the R groups of the amino acid. So the first type of bond that is formed in case of tertiary structure of proteins is known as the hydrophobic interactions or the hydrophobic bonds. So they are formed between the non-polar R groups. So the hydrophobic bonds are formed between the non-polar R groups, where most of these bonds are found usually in globular proteins, where the hydrophobic bonds are present in the inside of the globular proteins, exposing the hydrophilic proteins for their interaction with the water molecules. Now, the second type of bond present in tertiary proteins is the hydrogen bonds. So hydrogen bonds are basically formed between the polar R groups. So all those amino acids having polar R groups participate in the formation of the hydrogen bonds. Now, the third type of bond which is present in the tertiary structure of proteins are the ionic bonds or the electrostatic bonds. So these electrostatic bonds are formed between the charged amino acids. So the oppositely charged amino acids, like an acidic amino acid, will react with the basic amino acid to give electrostatic or ionic bonds. Now the fourth and the most important type of bond found in 3D structure of proteins are known as the disulfide bonds, and these bonds are formed between the sulfur groups of cysteine residues. So they are formed between the sulfur groups of cysteine, which is an amino acid. And disulfide bonds are one of the strongest bonds of the 3D structure of protein, which helps in stabilizing its structure. So we can see that hydrophobic interaction, hydrogen bonds, and electrostatic interaction are all present in the 3D structure of the protein. Therefore, they are most likely to be found in the tertiary proteins. So therefore, they will not be our answers. But our answer will be option 2, that is ester bonds, because ester bonds are formed between the hydroxyl group and the carboxyl group, and they are not very strong bonds. Therefore, they cannot help in stabilizing the three-dimensional structure of the proteins, and therefore they are least likely to be present in the tertiary structure of proteins. So our final answer is option 2, ester bonds. Hope you understood this well. Best of luck.